Hey everyone, and welcome to another deep dive with us. We're going to be talking about something pretty fascinating today.
Oh yeah, definitely.
For all you medical microbiologists out there, this one's especially for you guys.
Should be really interesting.
Yeah. So we're diving into the genus Rubrobacter.
That's right.
And our guide for this journey is going to be Volume 5 of the second edition of Bergey's Manual of Systematic Bacteriology.
It's a great resource.
Yeah, and so we're going to kind of go through and see what's so special about this this genus.
Yeah, it's not every day you find a bacterium that can like shrug off gamma radiation.
I know. That was like the first thing I read about these guys, and I was like,
Yeah.
okay, we have to do a deep dive on this. So where does this microbe fit in like the grand scheme of bacterial classification?
Yeah, so Rubrobacter belongs to the phylum Actinobacteria,
Okay.
which is a huge group. I mean, it's known for its diversity and complexity.
Oh, yeah, for sure.
We're talking about a phylum that has like everything from harmless soil dwellers to, the bacteria that cause some pretty serious diseases.
Yeah.
But even within this group, Rubrobacter stands out.
Okay, so what makes it like the rebel of the Actinobacteria phylum?
Well, so its unique characteristics initially led to its placement in the subclass Rubrobacteridae.
Okay.
However, scientists are, they're constantly questioning and refining how we understand the microbial world.
Right.
And Rubrobacter is no exception.
Yeah.
So research by Ludwig et al. In 2012 suggested that Rubrobacter might actually be distinct enough to warrant its own class.
Wow.
Called Rubrobacteria.
So it's like challenging the status quo even in the world of taxonomy.
Exactly.
That's pretty cool. So I guess before we get too far, can we just like take a step back and kind of paint a picture for everyone listening of what Rubrobacter even looks like?
Yeah, so picture this. A gram stain positive, non-motile, non-sporulating bacterium.
Okay.
Usually shaped like an irregular rod.
Okay.
So kind of like a tiny little wonky cylinder.
Gotcha.
And they're usually around 0.8 to 1.0 by 1.0 to 4.0 micrometers.
Okay, so tiny,
Tiny.
but kind of adaptable.
And as they age in cultures, they can sometimes even appear coccoid or like a sphere.
Interesting. And what about their environmental preferences? Where do they like to hang out?
So these guys are obligate aerobes,
Okay.
meaning they absolutely love oxygen.
Okay.
You won't find them in anaerobic environments.
Gotcha. And I'm guessing in the lab
Yeah, they're positive for catalase and cytochrome oxidase tests.
okay, so like easy to identify.
Yeah, which is helpful for us.
Yeah, for sure. So they need oxygen. We know what they look like. They've got some enzymatic activities.
Mm-hmm.
But what really sets them apart is,
Their resilience.
yeah, especially when it comes to gamma radiation.
Yeah.
That's a really interesting part for our medical microbiology listeners, too.
Exactly.
So to understand this remarkable ability, we kind of have to go back in time to the discovery of Rubrobacter radiotolerance.
That's right.
This species was first isolated from, get this, a hot spring
Yeah.
with high background radiation.
Imagine that.
It's like a delicate flower thriving in the desert.
Exact.
It's crazy. So, I mean, just how tough are we talking here?
So our radiotolerance has a shoulder dose of 6 kilojoules,
Wow.
meaning it can withstand that level of gamma radiation before you even start to see significant cell death.
That's wild. And what about their D10 value?
Yeah, so their D10 value, which is the dose needed to kill 90% of the population, is 10.0 kilojoules.
That's insane.
So they're definitely a rock star in the world of extremophiles,
Yeah.
which has piqued the interest of scientists across various fields.
For sure. So I guess kind of moving on, we know that they're tough, but how many different like members of this family are there?
Yeah, so currently we know of 3 different species.
Okay.
Our radiotolerans, which we already talked about, are Taiwanensis and R. zelenophilis.
Okay.
And like any family, each member has its own little quirks and characteristics.
Okay.
And we've summarized these in Table 316 of Bergey's manual.
Awesome. A table.
Yeah.
Perfect. So let's dive into some of those details and see what makes each species unique.
Sure.
So first up, I'm guessing they all like it hot, given their penchant for extreme environments.
You're right. Our radiotolerance prefers a moderately toasty range of 46 to 48 degrees Celsius.
Okay.
R. taiwanensis and R. zelenophilis crank it up a bit further, thriving at 60 degrees Celsius.
So we got 1 moderate thermophile and 2 extreme thermophiles,
Yeah.
all within the same genus. That's amazing. But what about their like dietary needs?
Yeah, so that gets into their biochemical and physiological characteristics, which are essentially their metabolic fingerprints.
Okay.
So, for example, our radiotolerance has a sweet tooth for glucose,
Okay.
while R. taiwanensis and R. zelenophilis can break down xylan,
Interesting.
which is a complex sugar found in plant cell walls.
Oh, Okay, so very different, but also similar.
Exactly.
So we're peeling back the layers of the onion here, finding more and more complexity.
And there's more.
Oh, there's more.
Yeah. So remember those unusual internal branched fatty acids we mentioned earlier?
Yeah.
Those actually play a crucial role in distinguishing these different species.
Oh, really?
Yeah. For example, our radiotolerance is rich in 12-methyl-hexadecanoic acid,
Okay.
while the other two species favor 14-methyl-octadecanoic acid.
Oh, wow. So it's like they each have their own unique fatty acid signature.
Exactly.
That's really cool. That's super useful for identification.
Very much so.
So this is all incredibly interesting, but I know our medical microbiology listeners are probably like, okay, how do we actually find these things
Right.
in the lab? Is it as easy as like swabbing a surface and hoping for the best?
Well, not quite. Isolating Rubrobacter can be a bit tricky because they're slow growers.
Okay.
It's kind of like finding a needle in a haystack. You need the right tools and a good understanding of their preferences.
Okay, so what are the secrets? What tools and techniques do we need?
Well, one of the most clever tricks is to use gamma radiation as an enrichment strategy.
Oh.
So remember how we talked about their radiation resistance?
Mm-hmm.
Well, we can actually use that to our advantage. By exposing an environmental sample to a dose of radiation, we essentially eliminate most of the other bacteria,
Oh, that makes sense.
giving Rubrobacter a competitive advantage.
So it's like a head start in the microbial marathon.
Exactly.
I like that. And then once we zap it with radiation, what happens?
Then we plate it on
Okay.
a specific type of media called thermos medium,
Okay.
which provides the optimal nutrients and conditions for them to grow.
Okay.
But even then, we need to be patient.
Gotcha.
We have to incubate these cultures at specific temperatures
Mhm.
and pH levels
Okay.
for optimal growth.
So patience, precision, and a really good understanding of their needs. It's
Exactly.
like building a little tiny extreme environment for them in the lab.
Exactly.
This has been an awesome deep dive so far.
Yeah, it has.
We've covered so much ground, from taxonomy to unique features to how tough they are to how to even grow them.
It's been great.
But before we wrap up, I actually want to give our listeners a little challenge,
Oh,
a
okay.
little exam to test your Rubrobacter knowledge.
I like it.
Are you ready?
Let's do it.
Okay, so first question. What makes the genus Rubrobacter so interesting for medical microbiologists? Think carefully.
Yeah, take your time.
The answer's in everything we've talked about so far.
Mm-hmm.
All right, question number 2. Describe the unique characteristic of the cellular fatty acids found in Rubrobacter species.
This one delves into the molecular details that make these bacteria so special.
Okay, on to question 3. What is the significance of the research by Ludwig et al., 2012, in the classification of Rubrobacter?
This one will test your knowledge of bacterial taxonomy
Yeah.
and its ever-evolving nature.
All right, question number 4. You are tasked with isolating Rubrobacter from an environmental sample.
Ooh,
What specific techniques would you employ and why?
this is where your practical skills come into play.
Exactly. And finally, question number 5. Compare and contrast the 3 known Rubrobacter species based on their key characteristics,
Okay.
emphasizing their differences in optimal growth conditions and metabolic capabilities.
So this one requires you to synthesize everything we've learned today.
Yes.
Mm.
Don't worry if some of these questions seem a little challenging.
Yeah, the goal is just to spark your curiosity
Yeah.
and encourage you to think critically about everything we've discussed.
Exactly.
We'll be back in the next part of the deep dive to discuss the answers in detail.
Yes, and explore even more fascinating insights about Rubrobacter.
Can't wait.
I know, it's going to be good. Welcome back to our deep dive into the world of Rubrobacter. I hope you had a chance to ponder those exam questions we left you with.
Yeah, a little mental workout is always good.
Definitely. So now it's time to unpack the answers.
Let's do it.
Solidify your Rubrobacter knowledge.
All right.
So let's start with that first question. What makes Rubrobacter so interesting for medical microbiologists?
Yeah, what is it?
I mean, for me, the answer is pretty clear. It's got to be their insane resistance to gamma radiation.
Yeah. I mean, you're not wrong.
Right. It's not every day you find a bacterium that can shrug off a dose that would completely fry most other microbes.
Exactly, and that's what makes them such a valuable resource for medical microbiology.
Yeah.
I mean, imagine what we could learn by studying their DNA repair mechanisms.
Right.
It could lead to new ways to protect human cells from radiation damage,
Oh, yeah, for sure.
from things like radiation therapy or even just like environmental exposures.
Wow. So it's like they have this biological shield that we can learn from.
Yeah, pretty much.
That's a really cool concept. Okay, let's move on to question number 2. Describe that unique characteristic of the cellular fatty acids found in Rubrobacter.
Yeah, so this one was all about the molecular details that set Rubrobacter apart.
Right?
So those internal branched fatty acids we talked about.
Yeah.
Most bacteria have fatty acids with branches at the ends,
Okay.
but Rubrobacter has them in the middle.
Oh, wow.
It's like they're doing things backwards.
The rebels even on a molecular level.
Yeah, exactly. And these unusual fatty acids might actually play a big role in their resilience.
Really?
Yeah, maybe by like adding some extra stability to their cell membranes, helping them withstand those harsh conditions.
That's awesome.
Yeah.
All right, question number 3. What is the significance of the research by Ludwig et al. 2012 in the classification of Rubrobacter?
Yes. So this one is about shaking up the bacterial family tree.
Exactly.
So Ludwig and their team proposed that Rubrobacter was unique enough to have its own class called Rubrobacteria.
Right, they're special.
Yeah, and it just goes to show that scientific classification is always evolving.
For sure.
We learn new things.
Yeah, totally. All right, question number 4. You're tasked with isolating Rubrobacter from an environmental sample. What do you do?
All right, time to put on our lab coats.
Let's go.
So first things first, knowing that Rubrobacter is a slow grower and extremely resistant to radiation, we can actually use that radiation resistance to our advantage.
Right, we talked about that.
Exactly. We can pre-treat the sample with gamma radiation.
So like a microbial cleanse?
Pretty much, it's like giving them a spa day while knocking out their competition.
I love it. And then what?
And then we plate it on thermos media.
Okay, give it the nutrients it loves and wait.
Okay, so patience is key.
Oh, yeah, definitely.
It's a waiting game. All right, last question. Compare and contrast the 3 known Rubrobacter species.
Okay, so for this one, you really got to know your stuff.
Right, we're bringing it all together. So when it comes to growth conditions, remember that our radiotolerance prefers it a little bit cooler.
Okay.
46 to 48 degrees Celsius. While the other 2 are Taiwanensis and R. xylanophilus, they like it hotter. 60 degrees Celsius.
Some like it hot, some like it even hotter.
Yeah, and then metabolically, our radiotolerance loves glucose,
Okay.
while R. taiwanensis and R. xylanophilus can break down xylan.
So they each have their own little food preferences.
Exactly.
It's really cool how these tiny differences can make such a big impact.
For sure. And don't forget about their unique fatty acid profiles.
Oh, yeah, those are important too.
Those are like their fingerprints, helping us tell them apart.
So cool. Congratulations, everyone. You made it through our Rubrobacter exam.
You did it!
You've officially earned your microbial merit badge.
And more importantly, I hope this has sparked your curiosity to keep exploring this amazing microbial world.
As we wrap up our deep dive into Rubrobacter, I can't help but think about, like, the bigger picture, you know?
Yeah.
Their resilience, it just makes you think about life in extreme environments.
Absolutely.
Even, like, on other planets.
Exactly. Their ability to survive in places that would be lethal to most organisms has huge implications for astrobiology.
Right. Like, could there be other extremophiles out there?
Oh, yeah, for sure.
Maybe even in places we thought were impossible for life.
It opens up so many possibilities.
It's crazy to think about. And then what about back here on Earth? Could we use Rubrobacter's adaptations for, like, bioremediation or even medicine?
Yeah, definitely. I mean, imagine using them to clean up radioactive waste sites.
Oh, wow.
Or using their DNA repair mechanisms to develop new drugs.
That'd be a video base.
Right. It's like taking inspiration from nature's ultimate survivor.
Yeah, and using it to solve some of our biggest problems.
Yeah, exactly. Rubrobacter is a perfect example of how basic research in microbiology can lead to all sorts of applications.
Totally. Well, this deep dive has been a journey for sure.
It has.
We've learned so much about Rubrobacter, but also how interconnected science really is. From taxonomy to biochemistry to astrobiology to medicine.
It's all connected.
And for our listeners who want to learn even more, I highly recommend checking out Bergey's Manual of Systematic Bacteriology.
It's an amazing resource.
It really is. So to all our fellow medical microbiologists out there,
keep exploring.
The microbial world is full of surprises.
It really is.
You never know what you might find.
That's the beauty of it.
Thanks for joining us for this deep dive into Rubrobacter.